Okay, good morning, everybody. I'm Gerald Soff. I'm the chief of the General Hematology Service at the University of Miami Sylvester Cancer Center, and I'm the course director and founder. We're going to have our first presentation today by me, and it's going to be the coagulation system. So traditionally, we've called it the coagulation cascade. I'm going to review the material at the level of a hematology specialist. We've all learned the classic Y-shaped cascade, and that's what we've memorized over the years. And I want you to, us collectively, to sort of get beyond that first level of understanding into the 21st century view of the coagulation system. A couple of housekeeping things. Thank you for registering, for those of you who have. At the end of the presentation, I will post the video link and the slide decks in the course homepage, Hematology Education Online. We'll also be sending out a survey for your feedback on how this is going. And there'll also be questions at the bottom for CME. And over the course of the next year, we will be able to get documentation and certification for compliance. So here's my disclosures. Personally, I received research support from Amgen, Janssen Pharmaceuticals, and Dover Pharmaceuticals, and I've participated in advisory boards from the following companies in the past 5 years. There's no commercial link to this content. And over the last couple of years, when I put this presentation together, I've received some feedback from some of my friends and colleagues shown here. So this is an overview of what we're going to cover. We're going to review the concepts of hemostasis and coagulation. We're going to talk about the classic coagulation cascade and the other topics here. I'm not going to read this because we're going to cover these as we go. So the concept of hemostasis, we use the term basically about bleeding, but in its pure Latin sense, it's keeping the blood liquid in the vasculature. We want to prevent hemorrhage following injury, and we want to also prevent excessive clotting in the vasculature. When we talk about hemostasis, we view it as primary hemostasis, which is a mixture of vascular forces, vasoconstriction, and platelet plug formation. And then we talk about secondary hemostasis, which is the coagulation factors leading to a fibrin clot. And for everything that turns on in the human body, we have to have things to turn them off. So we'll also talk a little bit about the physiologic anticoagulation processes. And this is just showing the same concept in sort of schematic form. So here's a cartoon of a vascular injury. Here's a small vessel, a laceration, and you can see the blood cells are pouring out. When this occurs, it's followed by vasoconstriction and platelet plug formation. So things that affect vascular tone and things that affect platelet number or function are classically thought of as affecting primary hemostasis. We're not going to discuss this today. That's going to come up in other talks. Following the platelet plug formation and exposure of the blood to subendothelial matrix, then you start the coagulation cascade, and that leads to a fibrin clot and cessation of bleeding. In reality, there's a lot of interaction between platelet plug formation and platelet function and the coagulation cascade. So we talk about them, we study them separately, but there is a lot of overlap. The earliest understanding of the coagulation system was attributed to Paul Marowitz in 1905 when he described 4 factors of the coagulation system. And we still use the terminology factors. We still use the terminology with Roman numerals. Fibrinogen or factor 1, that's the protein that fundamentally makes the clot. Prothrombin, in his concept, was an enzyme in the blood that converted fibrinogen to a clot. Now we know about thrombin derived from prothrombin. Thromboplastin factor, which we know is a mixture of tissue factor and phospholipid, and then calcium, they use, which was factor 4. We don't really talk about factor 3 and factor 4. We talk about tissue factor and calcium, but that's why when we talk about the clotting factors. There is no factor 3 and 4. The real development and breakthrough started in the 1930s when the prothrombin time was developed by Armin Quick. It was originally called the Quick time. And then in 1953, the partial thromboplastin time was developed. These tests have a lot of similarity. Basically, one takes platelet-poor plasma, adds thromboplastin or an activator. Thromboplastin would be for the prothrombin time, and an activator would be for the partial thromboplastin time. And then phospholipid, and then the one adds calcium. So I should mention that the plasma, in order to prevent it from clotting in the test tube, when it's collected and processed, would have a calcium chelator. So recalcification is the critical last step in activating coagulation in vitro. And then one would measure the time for a clot to form, and that would be either the PT or PTT accordingly. The early test was called the PTT, the partial thromboplastin time. Subsequently, it was modified, I think in the 70s, to add an activator. So that's where we call it an activated partial thromboplastin time. So these tests were developed and really were instrumental in clarifying that there were multiple clotting factors, because they would take plasma from a patient, and if it was prolonged, there was presumed to be a deficiency. And then what the hematologists did is they took plasma from a patient who had a deficiency, whatever it was, as an example, factor 8, and they would add plasma from a different patient with an unknown deficiency. And if the one-to-one mix corrected, then there was a concept of they were different factors because they could each correct each other. And that's sort of illustrated here. So we would take plasma from a patient with a known bleeding disorder, factor 8 or hemophilia A, and we would add plasma from an unknown, shown here, do a one-to-one mix. And basically, if the levels of any factor is about 50% or higher, the one-to-one mix would be normal. So if you had factor 8 and factor 9, the one-to-one mix would be normal. And that was the concept here. This worked really well. And from the 1930s to the 1950s, pretty much all of the clotting factors were developed this way. One of the limitations of the assay is that a range of inhibitors will interfere. And the use of the mixing studies today is for inhibitor measurement and titration. But the original purpose was to identify new clotting factor deficiencies. And in 1964, two seminal papers came out, basically both showing the origin of the coagulation cascade and the ordering of the factors. And both of them were very similar. They both showed, factor to factor to factor, one would activate the next, would activate the next, and you have this linear process. Earl Davey and Oscar Ratnoff called it a waterfall sequence. Robert McFarland from Great Britain called it a cascade, but the concepts were very similar. If you notice, both of them use Roman numerals, but also names, at least in the Davey-Ratnoff paper. And these papers have basically stood the test of time for decades and formed the fundamental understanding of the coagulation cascade. So what do we take from this? Number one is that the coagulation cascade involves a sequence of reactions. Factors circulate in a non-activated form, a zymogen or a proenzyme or a pro-cofactor. And we'll come back to this. The other important concept was that factors are activated in a proteolytic, by proteolytic cleavage by an upstream factor, and in turn activate a downstream factor. And the terminology became little a for activated. So for instance, factor 8 can be written this way, factor 8 or F8. We still use the Roman numerals, and that would be factor 8A or factor 8A, that's shown this way, and that would be illustrative of the activated factor. But there's a lot of gaps and questions, and we're going to tease these out and discuss these in the slides to come. So this is where we are now for the current understanding of the coagulation cascade. We have the intrinsic arm shown here in red. We have the extrinsic arm, and we have the common pathway shown here. And then we have factor 13 cross-linking. I'm showing at this very preliminarily, but we're going to tease this all out in greater detail. But one of the gaps in the original cascade is, where is factor 7, right? This is just a single arm. So the extrinsic arm was not present in these cascades. And there's other gaps as well, which we're going to talk about. So to get there, we're going to next talk about structure of the coagulation factors. And this is just showing the classic vitamin K dependent factors, factor 7, 9, 10, and prothrombin shown here. And they're all very similar in that they have certain conserved domains. GLA is gamma glutamic acid, gamma carboxyglutamic acid. We'll show that in a moment. They have other components here, EGF receptor, and these like domain, and these are conserved in most of the proteins. And then there's a domain here, which is the serine protease domain, and that has the enzymatic activity. But as these proteins are formed, they are proenzymes. They are not active. They have the capacity to become activated, but they're not yet activated. And we'll talk about this in a moment, but a series of proteolytic cleavages, sometimes one per enzyme, sometimes two, but they all require proteolysis to become activated. And this is just shown here as one example for factor 9. So the original factor 9, the inactivated, has this domain in the middle here and a disulfide bond here. But with activation, with cleavage, and there's two cleavage sites, an activation peptide is released. This is measurable in research laboratories. And then one is left with a 2-chain enzyme, a heavy chain and a light chain. The heavy chain contains the gamma carboxyglutamic acid domain, and we'll talk about this, but as you know, this is essential for calcium binding and formation of the activated enzyme complex. The heavy chain also helps dictate substrate specificity. The heavy chain has domains that bind to whatever substrate the enzyme will cleave. Then there's a disulfide bond here, illustrated SS here, linking it to the serine protease light chain. So the enzymatic domain is in the light chain, and that's pretty much conserved in all of the enzymes of the coagulation system. The enzymes are referred to as serine proteases because they all have serine and serine histidine and aspartic acid in the enzymatic pocket. So if one is looking at a gene for the gene sequence for a novel gene of unknown function, and you see a serine, histidine, and aspartic acid in this relative orientation, you could be fairly confident that that would be an enzyme, a serine protease. So that's the class of enzymes of the clotting of the coagulation system. They're called serine proteases because of the serine amino acid in that domain. Just like the enzymes have to be activated, the cofactors have to be activated. And tissue factor is one of the cofactors, but we're looking here at 5 and 8. And both of these also exist as pro-cofactors, requiring a series of proteolytic cleavages to become activated. So just like the enzymes require activation, the cofactors require activation as well. The structures look very, very similar, but they're actually on different chromosomes. The factor 8 is on the X chromosome and factor 5 is on chromosome 1. So probably in evolution, there was some genetic duplication which was preserved. But again, they have very similar domains, the A1, the A domain, B, and C, and then obviously there's subdomains within those three groups. Another critical concept, which I think every medical student historically has learned, is the role of vitamin K. So vitamin K is essential for a post-translational modification of the glutamic acid residues in many of the clotting proteins. So unmodified glutamic acid has a single carboxyl group at the end with a single negative charge. In a vitamin K dependent process, a second carboxyl group is added, and now you have a divalent anion in the amino acid in the enzyme, and that allows for calcium binding, a divalent cation. So vitamin K plays an essential role in post-translational modification and allowing these enzymes to bind calcium. Warfarin, for many, many decades, the only oral anticoagulant, works by blocking this pathway, resulting in which one is unable to form fully functional clotting factors. So there's two ways of initiating the coagulation system in vitro. And again, this is the classic teaching. We're going to understand as we go the limitations to this model. But this is how the intrinsic pathway gets activated in vitro, which is the blood, the anticoagulated plasma gets exposed to a negatively charged surface. Originally it was glass, and now there's chemicals that are added to the mix that has a negative charge. The result of that is factor 12 can become activated using prekallikrein and high molecular weight kininogen, and we'll tease this out in a moment, resulting in factor 12A. So this is how the intrinsic pathway gets activated in vitro. This is the beginning of the PTT. The extrinsic pathway gets initiated by addition of tissue factor, a protein enmeshed in phospholipid. So the terminology intrinsic pathway comes from the fact that if you add plasma to glass, it will clot intrinsic to the blood. Nothing is required to be added for this activation to occur. The extrinsic pathway gets its name because you have to add something extrinsic to the sample, which is a mixture of tissue factor and phospholipid. And this is the beginning of the prothrombin time. So we're going to talk briefly now about the contact phase, which is the beginning of the intrinsic arm. So the contact system contains three factors: factor 12, prekallikrein, and high molecular weight kininogen. And these form a complex on a negatively charged surface, shown here, and it becomes a positive feedback loop where factor 12 gets converted by kallikrein to factor 12A. Factor 12A can convert prekallikrein to kallikrein, so you have this positive feedback loop, and high molecular weight kininogen serves as a cofactor for these reactions to occur. The factor 12A can, as I said, feed back to activate more prekallikrein, but also can convert 11 to 11A, and this is how the contact system initiates the intrinsic arm of the clotting system. At the other end, at the bottom of the clotting system, of the coagulation cascade, we have the protein fibrinogen. Fibrinogen is a complex multimer of 6 peptide chains, 2 alpha, 2 beta, and 2 gamma, shown here in cartoon form. And it's a long molecule with these D domains at the end here and an E domain in the middle. The way fibrinogen gets activated is an enzyme, physiologically it's thrombin, sequentially cleaves parts of the alpha and parts of the beta chain, releasing what's referred to as fibrinate peptide A and fibrinate peptide B, and that results in a fibrin monomer, which is able to polymerize. The fibrin polymer, the fibrin monomers, which polymerize, are then stabilized across linked with factor 13A, shown here in cartoon form, and that results in a cross-linked stabilized fibrin clot. So that's how we see things at the bottom. And we've all learned about the D-dimer, just shown here in different cartoon formats. These are only released when cross-linked fibrin is degraded by fibrinolysis, by enzymes that degrade fibrin, and we'll talk about that later on in the presentation. So if one sees elevated D-dimer in a sample, that tells you that cross-linked fibrin has existed in the body. This could be from a clot, it could be from inflammation, it could be from a number of things, including cancer, but the D-dimer simply tells us that cross-linked fibrin has formed in the body and been proteolyzed. So that's sort of the basics of the coagulation cascade as it's been understood for many, many years. But we've also recognized in the hematology world that this understanding of the coagulation cascade really has some major inconsistencies and gaps. So for instance, we look at the intrinsic pathway, and I'll show that again in a second, but some deficiencies in the intrinsic pathway are associated with severe bleeding, and other deficiencies do not cause bleeding at all. And one factor in the middle, factor 11, causes moderate bleeding. So how can all these enzymes in one pathway have different manifestations? We've also understood now that this concept of each factor is in a cascade where they're activated by the enzyme above and activate the enzyme below is also incorrect. And we'll talk about that. And we now know that some enzymes have multiple substrates and some factors have multiple means of becoming activated. So we're going to talk about this because I think this is key to understanding the coagulation system as a hematology expert, not as a medical student. So to get at this, we're going to talk about the cell-based coagulation model. Rather than that nice Y-shaped picture of Roman numerals, which seem to be in isolation, that's not how the real body works. So this is an illustration of a cartoon illustration of a enzyme substrate cofactor complex. So in green here is a illustration cartoon of a membrane. There's a cofactor that anchors the complex. There's an enzyme that forms part of the complex and a substrate. So each of these complexes in the coagulation system has a cofactor, enzyme, and substrate. And I call this enzyme 1 because it cleaves the substrate, which becomes a new enzyme, enzyme 2, and that will then work at the next stage of the clotting system. So we're going to talk about this a little bit. And here are 3 enzyme complexes that represent what's actually happening in key stages of the coagulation system. So we have the extrinsic 10-ACE, which is simply a complex of tissue factors to cofactor, factor 7A is the enzyme, and factor 10 as the substrate. So this complex here of tissue factor, factor 7A, can then convert factor 10 to 10A. That's why it's called the extrinsic 10-ACE, because it cleaves 10 into 10A. In addition, there's the intrinsic 10-ACE, which uses factor 8A as the cofactor, factor 9A is the enzyme, and again, factor 10 as the substrate converting it to 10A. So this is where we've classically thought of the extrinsic and intrinsic pathways, each feeding in and activating 10, the beginning of the common pathway. And then you have factor 10, 10A as the enzyme of the prothrombinase complex, using factor 5A as the cofactor and prothrombin as the substrate. So this is shown here, factor 10A, 5A, and prothrombin as the substrate, and this is the common part of the clotting cascade. So understanding the cell-based mechanisms, the surface complex-based mechanism, helps put this Y-shaped schema into some biological sense. It doesn't fully explain everything, but we're getting there. So again, 3 key complexes you should remember: extrinsic 10-ACE, intrinsic 10-ACE, and the prothrombinase complex. Okay, now we're going to talk about the crossover, which starts to explain some of the gaps and misunderstandings. So, as I said a moment ago, the deficiencies of the contact system, factor 12, prekallikrein, and high molecular kininogen, even though they dramatically impact the partial thromboplastin time, do not result in bleeding. As a matter of fact, in the hematology world, it's famous that factor 12, named for Mr. Hageman actually died of a pulmonary embolism, not from a hemorrhage. Factor 11 deficiency is associated with a mild to no bleeding, but some of these patients will have mild bleeding, in particular post-surgery. And factor 8 and factor 9 deficiencies, the classic hemophilia A and hemophilia B, is associated with severe bleeding. So if 9 gets activated by 11 and 11 gets activated by 12, why would there be such a dramatic difference in severity based on the deficiencies of these factors? So to understand this, we have to now get the first real break from our concept of the classic coagulation cascade as a Y-shaped structure. Because reality is tissue factor 7A complex has 2 different substrates. We've all learned in the, studying the prothrombin time that this can activate 10, but in vivo, it also can activate factor 9, shown here. So tissue factor 7A activates 10 to 10A, but also can activate 9 to 9A. So this is the first example of sort of a crossover between the extrinsic and intrinsic arms. And so why is 9 important? Because if 10 can be activated directly, why would deficiencies of 9 or its cofactor 8 be clinically relevant? Well, what the key here is tissue factor pathway inhibitor, TFPI. And what we understand now is that in the body, TFPI dramatically blocks this reaction, dramatically reduces the ability of 7A to activate 10 to 10A, leaving its primary substrate in the body in vivo as factor 9. So this is what the coagulation system actually looks like. We have factor 7 theoretically has the ability to convert 10 to 10A. In vitro, it works fine, but in vivo, in the body, it's dramatically reduced. Therefore, the 7A tissue factor complex activates 9 to 9A, and using 8A as its substrate, this is how most of the coagulation activity occurs in the body. And that's why in the absence of 8 or 9, there's severe bleeding, even though theoretically this pathway could occur. This is exploited pharmacologically in that there's recombinant 7A available for patients with inhibitors to factor 8 or factor 9, and that will overcome this. But physiologically, this is what's actually happening in the vast majority of situations. And that explains why 8 and 9 deficiencies are severe, but 11 deficiency in the contact system is less relevant. So this is just showing the same concept again. This is what the real cascade looks like in the body. The next concept we want to talk about is the thrombin burst. And this is there's some fun illustration of a burst, of a gamma burst from something exploding in space. Because this is going to address a lot more gaps. One of them is like, how does factor 5, 8, 11, 13 get activated in the classic cascade? And the answer is we don't know. But now we understand that there's a thrombin feedback, and we're going to talk about that. So the term thrombin burst is used to describe a dramatic augmentation of the coagulation system once some thrombin has been formed. And that's a series of positive feedback processes. And that's illustrated here. So here's thrombin formed, it's in gold. And rather than have all the arrows feeding back, which would make this illustration a mess, I'm just showing here with these horizontal arrows, all the different stages in the clotting system where thrombin feeds back to further activate clotting factors. So thrombin, or factor 2A, because prothrombin is 2, right? So thrombin is factor 2A, activates 11 to 11A, also activates the cofactors 5 and 8 to 5A and 8A, cleaves fibrinogen, which is its oldest known function, and also activates factor 13 to factor 13A, and that leads to cross-linking of the fibrin clot. So thrombin has a whole series of positive feedback contributions to the coagulation system. This also explains why patients with 11 deficiency have some bleeding, because 9 and 8 can get activated by 7A, as we talked about, but much of the without the role of factor 11, but factor 11 can be activated as part of a thrombin burst and feedback and further activate additional factor 9. So these are some of the activities, procoagulant activities, and In addition, I put it in brackets because it's not a clotting factor, but thrombin can also feed back and activate platelets. Okay, so this is just showing the role of factor 11 here. It's a component of a positive feedback loop. Here's thrombin, and I do have one of these feedback arrows showing 11A gets activated, can further activate factor 9. But it also illustrates why these components here remain superfluous in most in vivo settings of clotting. So next thing we want to talk about is the hemostatic balance. So we've talked about our current understanding of the physiologic processes leading to coagulation. Now we're going to talk about the physiologic anticoagulation processes. And it's a balance, right? If there's too much coagulation, one has thrombosis. If there's too much, if there's a deficiency of the coagulation factors, we have hemorrhage. And if there's deficiencies of the physiologic anticoagulation, that also leads to thrombosis. So here is the vasculature illustrating its contribution to the physiologic anticoagulation processes. So this is illustrating intact endothelium with anticoagulation surface shown here and here. If you have a gap in the endothelium, platelets adhere, and that starts the coagulation system. But where the endothelial cells are intact, you do not want the blood to clot. So the endothelial cells are the adjudicators, so to speak, on whether the blood is going to clot or not. And there's a range of factors, a range of processes on intact endothelium that inhibit or actually reverse the procoagulant state. Endothelial cells do not activate clotting, which is very important. But beyond that, they actually regulate and reduce the rate of clotting shown here. So there's a lot of these pathways which we're not going to talk about. There's a CD39 or ADP ecto-ADPase that degrades ADP, and as we'll talk about later in the year, ADP activates platelets. Endothelial cells synthesize nitric oxide. Nitric oxide also inhibits smooth muscle contraction and also inhibits platelet function. Cyclooxygenase reduces smooth muscle tension and inhibits platelet function. So these all work to inhibit platelets, which we're not going to discuss today. There are two critical pathways that are endothelial cells dependent that turn off the coagulation system: heparan sulfate using antithrombin, and the thrombomodulin protein C, protein S pathway, which we'll talk about in a few moments. And we already talked about tissue factor pathway inhibitor that blocks the direct conversion of factor 10 to 10A by tissue factor 7 complex. So this is endothelial based, and that's why it only works in vivo, not in vitro, because you don't have endothelial cells there. So we're going to first talk about anti-thrombin glycosaminoglycan. So antithrombin is a protein that has the potential to neutralize the enzymes of the coagulation system. However, as antithrombin circulates, it does not bind or neutralize enzymes. This is heparan sulfate, which is a glycosaminoglycan anchored to an endothelial cell here. You can see it looks like a tree in a way. And this is the core protein, and these are the active side chains, the glycosaminoglycan side chains. This is heparan sulfate, and it's chemically very similar to heparin sulfate, as you all know, a very important drug. So in the natural state where the blood is flowing, you do not see any activity here. However, or should I say, so how does this work? That if there's binding of antithrombin, also called antithrombin 3, to glycosaminoglycans, it undergoes a steric alteration takes on the conformation, allowing it to bind to heparin or other enzymes in the clotting cascade, and therefore it neutralizes the enzyme and dissociates from the heparan sulfate, and the complex is then removed from the circulation in the liver. So just to recapitulate, under physiologic conditions, antithrombins in the flowing, it doesn't do anything. When it binds to heparan sulfate or glycosaminoglycan, it becomes activated, or a conformational change, I should say, allowing it to bind to and neutralize the enzymes in the clotting system. And then those are cleared. The second pathway is the protein C, protein S thrombomodulin system. So the heparan antithrombin system neutralizes the enzymes in the clotting system, in the coagulation cascade, and this pathway neutralizes the cofactors. So here's the different constituents, but I'll show you two in cartoon form. It's easier to see. So again, when the thrombin is in circulation, it has the ability to convert fibrinogen to fibrin and other procoagulant activities. So this is its baseline activated activity. However, in the presence of intact endothelial cells, there's a protein called thrombomodulin shown here, anchored to the endothelial cell. And thrombin will then bind to thrombomodulin shown here, and the thrombin is modulated or undergoes a conformational change itself. And when thrombin is bound to thrombomodulin, it no longer has the ability to cleave fibrinogen to form fibrin clots, but now it takes on a novel spectrum of enzymatic activity, and it becomes an anticoagulant. So this is the first step here. Thrombin sees thrombomodulin on an intact endothelial cell, it binds, and it undergoes a conformational change. When thrombin is bound to thrombomodulin on the endothelial cells, it also then gets co-localized to protein C. So this is the endothelial cell protein C receptor, or EPCR. And the EPCR does what its name implies, is it binds protein C, allowing thrombin now to convert protein C to activated protein C, shown here. So again, when the endothelial cells are intact, thrombin is modulated by thrombomodulin and activates protein C to activate protein C. Protein C then uses its receptor protein, its cofactor protein S, and will then cleave and inactivate factor 5A and factor 8A. So this pathway here with thrombomodulin, endothelial protein C receptor, activated protein C, protein S will then neutralize factor 5A and 8A. I didn't put it in here, but there's also believed to be some other roles of the protein C system, possibly fibrinolysis, but this is considered to be the primary role of the protein C pathway. The last of the physiologic processes, last of the physiologic anticoagulation processes affecting the clotting factors, is the fibrinolytic pathway. And it has a range of components listed here, but again, we'll tease this out individually to help you understand it rather than just try to memorize everything. So this is the clot-based fibrinolysis that occurs. So plasminogen, this is an illustration, cartoon form of a fibrin clot. So plasminogen can bind to fibrin. TPA, or tissue plasminogen activator, can bind to fibrin. And when they both bind to fibrin, plasminogen gets converted by TPA to plasmin. So this is a proenzyme or zymogen. Here's the active enzyme. Plasmin, when bound to fibrin, would be like a Pac-Man. It chews it up, and then you have fibrin degradation products. So this is the core of a clot-based fibrinolysis, a clot-based process by which fibrin clots are degraded. There's an alternative way of activating plasmin, though. So here's showing a cell-based mechanism, plasminogen shown here, and then a range of cells, such as prostate cells and some other cells, uterine cells, will have UPAR on the surface, or urokinase plasminogen activator. And urokinase plasminogen activator will bind urokinase, or UPA, and activate it. So this is an alternative mechanism of activating, forming plasmin separate from a fibrin clot. So here, plasminogen does not have to be anchored to fibrin, but it can still become activated, forming plasmin, and the plasmin then has the ability to bind to a fibrin clot and lyse it. So there's two alternative pathways for fibrinolysis to be generated. And for every inducer, every activator, there's always inhibitors, just like we talked about for the heparan antithrombin and the protein C, protein S thrombomodulin system. So just as we have plasminogen has the ability to get converted to plasmin, and there's two inhibitors of plasminogen activators, Pi1 and Pi2, or plasminogen activator 1, plasminogen activator inhibitor 2, shown here. And then there's a plasmin inhibitor, alpha-2 antiplasmin. And there's deficiencies, rare, but there's deficiencies pretty much all of these components. There are deficiencies of plasminogen. There's deficiencies of the Pi1 and Pi2 and alpha-2 antiplasmin. And as you can imagine, an alpha-2 antiplasmin deficiency would be resulting in excessive plasmin activity and bleeding, and deficiencies here would result in excessive plasmin formation as well, and leading to a hemorrhagic state. So that's what we have for now. I've tried to give you guys more than just a simple overview of the cascade. I want you to try to understand some of the key concepts as we talked about. And for instance, the differences in the factors in the intrinsic arm, and the roles of some of these physiologic inhibitors, as well as the role of the thrombin burst. Understanding this will go a long way to allowing us to understand, approach the patient better.